Hello, my name is Hane Lee, and I'm Chief Genomics Officer at 3billion. Thank you for inviting me to the conference. Let me go to the next slide. I'd like to thank the organizers for inviting me to give this talk. I wish I could have been there in person, but this is still a great opportunity for myself and 3billion. So we are thankful that the virtual option was available. Today, I will be talking about my experience of using genomic sequencing for diagnosing rare neurodevelopmental disorders. Here's the outline of my talk. I will start off with a few slides of introduction to genomic sequencing. Then I'll share a couple of cases that were diagnosed using exome sequencing and end with concluding remarks. First, the introduction. Just to make sure everyone's on the same page, let me briefly walk you through various terminologies. The central dogma of molecular biology is that DNA is transcribed into RNA, and RNA is translated into protein. When we say genomic sequencing, it usually means sequencing the genomic DNA, either the whole genome or a subset of the genome, such as the exome. Genic region is about 25 to 38% of the genome, but large portion of a genome is intron, the region that gets spliced out. Exome is about 1% of the genome, and it is the collection of the exons that gets transcribed into RNA. Depending on which exome capture kit is used, the untranslated regions, commonly called UTRs, could be included in the exome or not. Then there's transcriptome sequencing, which is commonly referred as RNA-seq, that sequences the collection of the messenger RNAs that were expressed in the tissue sampled. RNA-seq is being reported to be essential in interpreting variants from genome sequencing. Today, we will mostly talk about exome sequencing, but I will share a case towards the end that was solved by whole genome sequencing integrated with transcriptome sequencing. Genomic sequencing became available because of the next generation sequencing, or NGS technology. As opposed to Sanger sequencing shown on the left, that is performed to read a sequence up to 600-700 base pair or small region of the genome one at a time, NGS reads millions of short sequences simultaneously. Also, for NGS, there is no need to design specific primers for each fragment being sequenced. Many of you probably saw this figure on the right bottom that shows how the sequencing performance in terms of the cost has been exceeding the Moore's law. With the advent of NGS technology, the sequencing cost per base has dramatically reduced over the years. And today we can sequence a whole genome at less than $1,000, and it takes only about 3 days to generate enough data for a meaningful analysis. However, cost reduction is not the only reason genomic sequencing has become so popular. Here are some of the additional key reasons. First, as you can see in these graphs, the novel gene discovery rate has significantly increased as exome sequencing was introduced, impacting both the research and clinical communities. Second, as exome sequencing started to be offered as a clinical test a decade ago, it has made possible to finally diagnose patients who have been on the diagnostic odyssey for a long time by surveying all possible genes without having to select a few genes based on the phenotypes. The diagnostic rate from exome has also been relatively high at around 25% and up, depending on the disease domain, with a quite fast turnaround time compared to other more traditional tests, making exome attractive. Finally, even though precision medicine or personalized medicine has become a popular term only recently, if you think about it, clinical geneticists like many of you have been already practicing precision medicine in the rare disease world for a long time by finding the molecular diagnosis and managing the patients accordingly. I think advancement in technology to make a quicker and more accurate molecular diagnosis is just making precision medicine for rare diseases more robust. Now, let me share a couple of internal cases that were diagnosed using exome sequencing. But before I go into the cases, let me briefly explain our exome sequencing workflow from specimens to clinical reports. The entire workflow has been validated as one whole test. I would like to also mention that our laboratory is accredited by the College of American Pathologists, CAP. Currently, we accept whole blood and buccal swab samples. We also accept high-quality genomic DNA extracted from whole blood or buccal swabs. Once the genomic DNA is extracted and quality checked, library preparation and exome capture are performed. At 3billion, we're currently using a commercial kit from Integrated DNA Technologies, IDT, for capture. Sequencing is performed on Illumina NovaSeq, and we run 150 base pair paired end sequencing. The mean depth of coverage is around 100x, and more than 98% of the targeted bases are covered at least 20x. Once the sequencing is complete, the reads are aligned to the human reference genome and variants are called. We adopted GATK best practices for the variant calling pipeline. The variants are then annotated, classified, and filtered using our internal system called Evidence. We follow the ACMG guideline for variant classification, but we further customize it to make it more robust. We also update all databases as soon as there is a new release to make sure we are using the most up-to-date information for variant interpretation. Since we're searching for causal variants for rare disorders, all the common variants get filtered out, and the remaining rare variants get sorted by their symptom similarity score. Finally, our variant interpretation team will review the final list of rare variants and report out the causal variant. So here's case number 1. Case 1 was an 11-year-old Latino and mixed American male with a phenotype of intellectual disability and dysmorphic features. Karyotyping and CMA were performed prior to Exome, and they were basically normal or negative. There were no differential diagnoses suspected by the clinician. Exome was ordered as a proband only without the parental samples, and an extracted DNA sample was received. Sequencing quality was good, with almost 99% of the targeted regions covered well. We predict the karyotype from the sequencing data, and when it's not consistent with the gender information provided in the order, we will investigate. For example, here's a case for which the provided sex was female, but the data was inconsistent. At many times, this discrepancy turns out to be a mistake on the order, but this case was not that case. Our data suggested that the patient was mosaic, with about half the cells being 45XO and the rest being 46XY. There was no note from the physician, so we contacted him to be told that the patient was indeed a mosaic. The degree of mosaicism was a bit off, but that could have been explained by testing on different tissue type. For this case, it matched pretty well. And we also check for regions of homozygosity, and the reason is so that we can detect consanguinity, large deletion, and or uniparental isodisomy. Although this case did not have any regions of homozygosity, if there were some regions of homozygosity, you will see something like this. This is for a case for which the parents were known to be cousins. From the exome data, we could tell the parents were closely related cousins, probably first cousin once removed. So for this patient, all the QC checked off, so we moved on to the analysis. First, this patient opted in for receiving secondary finding information, but there were no pathogenic or likely pathogenic variants in the 73 ACMG recommended secondary finding gene list. So at the bottom here is a snapshot of the report describing the results. Then for the main phenotype, we found a heterozygous likely pathogenic missense variant in the SMARCA2 gene that is associated with autosomal dominant Nicolaides-Baraitser syndrome. This is a snapshot of the results section of our final report. We describe in the interpretation section why we classify the variant as likely pathogenic, and I'll walk you through this table. First, this variant is extremely rare as it is not observed in any of the large population database, such as NOMAD. Missense changes are a common disease-causing mechanism for this gene, and the variant is located in a mutation hotspot, as you can see here. This is a screenshot of NOMAD showing the ClinVar summary track. The red points are all the ClinVar pathogenic or likely pathogenic variants, and you can see that almost all of them are triangle, meaning they are missense variants. This is a pretty large gene, but there are certain regions these red triangles are clustered, and our variant is within one of those clusters. This figure pretty much shows the same thing. The disease-causing variants are clustered in specific domains, and our variant is in one of those important domains. Here, the segregation data says none, because this was a proband-only case. If this were a trio case and the variant was found to be de novo, it would say that. Next, as this is a missense variant, in silico prediction on how damaging this variant could be was calculated, and both Rebel and 3CNet, our in-house developed tool, predicted it to be quite damaging. I want to make a comment, though, that these predictions are only predictions, and we do not filter out variants based on these values. Next, this variant has been observed in other patients with similar phenotype, and a different amino acid change at the same codon has also been reported in patients with similar phenotype. So we can see both in ClinVar. Finally, most importantly, the phenotype. Again, I'm showing you what the physician gave us for the patient's phenotype on the top, and below is a snapshot from a paper describing this syndrome. We determined that it was a good fit for our patient, but some of you may be wondering that these phenotypes that our patient has are not so specific. That is true. It would be best if we can have more detailed information, even access to the patient's chart, but unfortunately, as a reference lab, that's almost not possible. So we have to combine the variant information with the phenotype information to make the final call. And here, since we have such a strong variant in a gene that could fit very well, we determined this variant to be disease-causing for this patient. So back to the results section, we reported out the variant as likely pathogenic, recommending parental testing to be performed to confirm if the variant is de novo or not. Now, the second case. Case number 2 was a 3-year-old South Asian male. Phenotype included global developmental delay with regression, hypotonia, and dystonia, increased lactate level, and abnormal brain imaging finding on MRI. The only genetic testing done prior to Exome was sequencing delay syndrome variants on the mitochondrial genome, which came back negative. The reason this test was done was because Leigh syndrome was on the differential. Exome was ordered as a proband-only case using extracted DNA sample. QC was good, and there was no secondary findings. For this patient, we found a pathogenic variant in MORC2 gene. It was a missense variant that has been reported multiple times in other patients with overlapping phenotype. This gene is known to cause a syndromic developmental delay disorder, but not a known mitochondrial disorder. So do we have the right diagnosis here? Let me walk through the variant information. Like before, this variant was not observed in nomads, so it's extremely rare. Missense changes are a known disease-causing mechanism, with all the ClinVar pathogenic, likely pathogenic variants being missense. This was also a proband-only case, so there's no segregation data. In silico prediction score was high by 3CNET, but not so high by Rebel. But again, we don't filter out any variants by these scores. This exact variant has been reported multiple times as disease-causing, and here are some of the entries in ClinVar. They were all submitted as pathogenic. And they have been reported as de novo in all the cases for which parental samples were available, as you can see here. This is from a paper. And again, most importantly, does the phenotype fit? In OMIM, we can see that this gene is associated with 2 different disorders, CMT type 2Z and developmental delay with impaired growth, dysmorphic facies, and axonal neuropathy. CMT was reported first from a handful of patients, but some of the patients diagnosed with CMT were reported to have additional phenotypes, as you can see here highlighted in yellow. So the authors of this paper, who first described this developmental delay syndrome, took a different approach. They collected all the patients with de novo variants in MORC2 and thoroughly assessed their phenotype to find common feature. So it's a genotype to phenotype approach. On the left is the patient's phenotype, our patient's phenotype, and on the right is the summary table from this paper. And you can see that most of the patients had more than CMT, with many of the symptoms overlapping with our patient's phenotype. Also, to note is that many patients in the paper had Leigh-like syndromes as well. And if you remember, our patient also had these symptoms and even received a genetic test for Leigh syndrome. The authors of this paper suggested that the variable phenotype associated with this gene may depend on the severity of the variants. Variants that exert stronger hyperactivating effects on the human's like silencing hub-mediated silencing may result in more severe CNS phenotype. And the variant that our patient has was studied to have a very strong effect. So in conclusion, well, we thought the phenotype fit very well. So we reported this variant as a pathogenic variant and recommended additional phenotyping and parental testing. So hopefully, I hope you found these 2 illustrative cases to be informative, convincing you that sequencing the entire exome and searching for the molecular diagnosis in an unbiased manner is preferred to performing a focused single gene or panel test, especially for heterogeneous disorders. Let me now make a few concluding remarks. This is the overall diagnostic rate of exome sequencing reported from large reference laboratories in the US. It hovers around 25 to 30%. This number has stayed remarkably consistent over time and across different laboratories. Of course, there are reports presenting higher diagnostic rate, but most of them are from smaller cohorts selected for certain disorder categories. Also, at least in the US, exome is still not the first tier test for various reasons. So I would say a lot of easier cases get diagnosed by single gene or panel tests first. The diagnostic rate does vary by disease domain though. So neurodevelopmental disorders tend to have a higher diagnostic rate, as you will see in the following slide. This is 3billion's exome diagnostic rate. Our overall diagnostic rate is around 45%, slightly higher. Why is ours higher than that from other larger laboratories? One possibility is that many of our collaborators are ordering our exome as the first tier genetic test, which I think is the right approach. The diagnostic rate varies by disorders though, but neurodevelopmental disorders tend to have a pretty high diagnostic rate, near 50%. However, there are more than half the patients still undiagnosed. What could be the reasons? Here are a few possibilities. First, it's possible that certain patients don't have a genetic disorder. Secondly, the causal gene may have not been discovered yet as a disease gene. It's also possible that certain variants are not identifiable by the current NGS technology or interpretable by current bioinformatic tools. If it's the second case, a reanalysis of the existing exome data will find a causal variant someday once the causal gene becomes a disease gene. And that's why reanalysis of the exome negative cases is critical. And 3billion actually runs reanalysis daily at no additional cost because of that reason. In summary, this is a pretty common workflow that is practiced for diagnosing rare neurodevelopmental disorders, although I think it can be easily applied to other disease domains. For a patient with suspected rare neurodevelopmental disorders, unless there is a highly suspected gene to test first, chromosomal microarray is usually performed first to find large copy number variants. The diagnostic rate for CMA hovers around 10%. If CMA comes back negative, large gene panels or exome sequencing is usually the next option. The diagnostic rate from exome is reported to be around 25%, although depending on the disease domain, it can go up to about 50%. For negative exome, it is now a common practice to wait a year or two and get the exome data reanalyzed because the diagnostic rate from reanalysis is quite significant at about 10%. Most of the increase comes from novel gene discoveries, as I said before. If still negative, whole genome sequencing is usually the next step if available. Whole genome sequencing can identify large structural variants missed from CMA or exome, and therefore increases the diagnostic rate up to about 10%. However, all the non-coding variants are still left in the dark, and that's where transcriptome sequencing comes useful. When whole genome and transcriptome sequencing data are interpreted together, we can identify cryptic variants that affect splicing or expression. So altogether, we can reach about 50% diagnostic rate. But of course, if exome diagnostic rate was almost at 50 already, it would go even higher. What happens to the rest who are still undiagnosed at that point? More novel gene discoveries and further advancement in technology to identify and interpret variants that are being missed or left uninterpreted will hopefully help solve them in the near future. One more thing I want to note here is that whole genome sequencing can actually replace CMA and exome altogether and find additional variants the two tests cannot identify. So once the whole genome cost is further reduced, it will be reasonable to just perform whole genome sequencing from the start and further reduce the time to diagnosis. 3billion is also heading to that direction and preparing accordingly. Let me briefly describe a real case that was diagnosed by whole genome sequencing integrated with RNA-seq. The patient was a 3-year-old male with global developmental delay with regression, seizures, and optic atrophy. Prior genetic testing was negative, and whole genome sequencing through the US Undiagnosed Diseases Network was negative for any structural variants that could explain the phenotype. We performed RNA-seq from blood sample on the trio and inspected rare genetic variants to search for alterations at the transcript level. And we were able to identify compound heterozygous variant in SLC25E46 gene that is associated with autosomal recessive lethal congenital pontocerebellar hypoplasia. The variants found were a maternally inherited missense variant that was not known to be pathogenic, and a paternally inherited 114 base pair deletion in the deep intronic allorepeat region that led to 2 different splice alterations, the inclusion of a pseudoexon and intron retention, right upstream of the deletion. Both proband and the carrier father showed the same transcriptional changes. So the patient was finally diagnosed with these findings, and this shows how significant the RNA-seq can be to interpret whole genome data. Now let me wrap up by proposing how clinical genetics practice could or should be done. This is how the current practice is mostly done. A patient is presented with a rare or common disorder, and the physician takes clinical history of the patient. Additional phenotypic data are collected by various tests, which are often expensive and frequently do not lead to a clear specific diagnosis. Genetic evaluations are usually ordered at the end to confirm the clinical impression, and at many times, multiple single gene or gene panel tests have to be ordered while still not leading to a clear diagnosis. In general, except for certain less heterogeneous disorders, the success rate of molecular diagnosis is quite low, and accordingly, ultimate costs to establish these rare diagnoses are high. This is how we envision how the future medical genetics practice will change to. If one or more genetic causes are highly suspected as the proximate cause of the symptoms, clinical exome or even whole genome sequencing will be ordered as a first-line test to establish the genetic diagnosis. Additional patient phenotyping will be guided by the genetic assessment and only ordered when necessary. So finally, I'd like to thank all our collaborators all over the world and their patients. Since 2017, we've sequenced and diagnosed more than 22,000 patients with suspected rare genetic disorders, and the number is continuously growing. I want to note that we've received a lot of samples from Egypt. I'm assuming many of the referring physicians are in the audience, so thank you. We're especially thankful for all the detailed clinical information given at the time of the order, including patient's photo, and they were very helpful. For your information, the diagnostic rate has been quite high for your patients at 44% with clear diagnosis. And in addition, 20% of the patients received inconclusive results. And some of these are actually patients with CNV results. So basically positive, they just need to be confirmed by alternate tests. And finally, I'd like to acknowledge our 3 billion team. We work hard not only to find the diagnosis, but also to further streamline and speed up our variant analysis pipeline and detect more complex variants. Here's my last slide. Thank you for listening.